The last module on viral pathogenesis looks at patterns of infection. And essentially, we're just looking at variations of two modes, either acute or persistent infections. An acute viral infection is characterized by rapid onset of disease, a relatively brief period of symptoms, and resolution within days. It is usually accompanied by early production of infectious virions and elimination of infection by the host immune system. You can see in this illustration here, the purple indicates the point where the innate defenses have been overwhelmed and the adaptive immune system had to kick in. Acute viral infections are typically observed with pathogens such as influenza virus and rhinovirus. Ebola hemorrhagic fever is an acute viral infection also, but the course of the disease is usually severe. So whether a disease is very, very severe or not, that has no effect on whether it's an acute infection. Often, an acute infection may cause little or no clinical symptoms, the so-called inapparent infection. A well-known example of this is poliovirus infection. Over 90% are without symptoms. During an inapparent infection, sufficient virus replication occurs in the host to induce antiviral antibodies, but not enough to cause disease. Such infections are important for the spread of infection because they are not easily detected. For instance, during the height of the polio epidemic in the U.S. in the 1940s, the quarantine of paralyzed patients had no effect on the spread of the disease because 99% of the infected individuals had no symptoms and were leading normal lives and spreading infection. Inapparent infections probably are most important features of pathogens that are well adapted to their host. They replicate sufficiently to ensure spread to new hosts, but not enough to damage the host and prevent transmission. Acute viral infections are responsible for epidemics of disease involving millions of individuals each year, such as influenza and measles. When vaccines are not available, acute infections are difficult to control. Most are complete by the time the patient feels ill and the virus has already spread to another host. This characteristic makes it extremely difficult to control acute infections in large populations in crowded areas like colleges and nursing homes. So let's take a look at measles. So let's talk about the measles virus. It's estimated that there are around 10 million cases annually with at least 100,000 deaths. The incubation period is about a week. The site of initial infection is the respiratory tract. It then moves on to local monocytes and lymphoid cells that migrate to lymph nodes. This is the primary viremia. The secondary viremia includes replication in epithelial cells of the lung, mouth, and then the skin. Key symptoms include cough, fever, and conjunctivitis. Now you'll notice that virus shedding occurs during the prodromal period, that is, before the symptoms are actually evident, particularly long before people have developed the rash. So that makes it much easier for the virus to be passed on from person to person. Persistent infections result in the continuous production of infectious virus, and this is achieved either by the survival of the infected cell or by a situation in which a minority of cells are initially infected and the spread of virus is limited, so that cell death is counterbalanced by new cells produced by cell division, that is, no net loss. Persistent infections result from a balance struck between the virus and its host. As an example, simian virus 5 or SV5 causes acute infection in BHK cells, but a persistent infection in monkey cells, or MK cells. The virus multiplies at the same rate in both cell types, but the MK cells show no CPE, remain healthy, and produce virus continuously. The BHK cells all die. Some persistent infections are known as latent infections. In a latent viral infection, the virus remains in equilibrium for the host for long periods of time before symptoms again appear, but the actual viruses cannot be detected until reactivation of the disease. Examples include herpes simplex 1, or the fever blister example that we talked about, where the virus is latent in neurons. Herpes simplex 2, which causes genital herpes, and VZV, varicella zoster virus, which causes chickenpox and then is reactivated as shingles. In the case of chronic virus infections, the virus can be demonstrated in the body at all times, and the disease may be present or absent for an extended period of time. Examples include hepatitis B and hepatitis C. Slow infections are ones in which the infectious agents gradually increase in number over a very long period of time, during which no significant symptoms are seen. Examples include AIDS, which is caused by HIV, and other kinds of lentiviruses that can cause tumors in animals. Although not viruses, prions also cause slow infections. There is no single mechanism responsible for establishing a persistent infection. A key feature is reduction in host defenses and the ability of the virus to kill cells. In some persistent viral infections, there are alternate cycles of virion production and quiescence. An example is Epstein-Barr virus, the agent of infectious mono. After the initial bout of fever, sore throat, and swollen lymph glands, the virus establishes a dormant infection in which the viral genome persists in the cells of the immune system. Periodically, the infection is reactivated and infectious virions are shed in the absence of clinical symptoms. These reactivations lead to transmission of the infection to new hosts. Many infections persist because viral replication interferes with the function of cytotoxic T lymphocytes, or CTLs, the immune cells that are extremely important for clearing viral infections. Infected cells are recognized when CTLs detect viral antigens on the cell surface. This recognition process requires presentation of the viral peptides by the major histocompatibility complex, or MHC, class 1 proteins. Many viral proteins interfere with different steps of the MHC class 1 pathway, including the synthesis, processing, and trafficking of the protein. Even transport to the cell surface of viral peptides can be blocked. An example of such immune modulation occurs in cells infected with cytomegalovirus, or CMV. The beta herpes virus causes a common childhood infection with little consequence in healthy individuals. The infection, however, is never cleared, and the virus persistently infects salivary glands, mammary glands, and the kidney. When latently infected individuals are immunosuppressed by drugs or HIV infection, viral replication ensues with life-threatening consequences. CMV persists in the host because the viral genome encodes multiple proteins that interfere with MHC class 1 presentation of viral antigens. One viral protein blocks translocation of peptides into the lumen of the endoplasmic reticulum, while two other viral proteins cause degradation of MHC class 1 proteins before they reach the cell surface. HIV attacks immune system cells in the body and uses the cell's machinery to make copies of itself. However, some HIV-infected immune cells go into a resting or latent stage. While in this latent stage, the infected cells don't produce new HIV. The virus can hide out inside these cells for years, forming a latent HIV reservoir. At any time, the cells in the latent reservoir can become active again and start making more HIV. There are many more examples of how virus infections modulate the immune response, leading to persistent infection. Not surprisingly, many of the processing or regulatory steps that are targets of viral modulation were not even known until it was discovered that they were blocked by virus infection. Okay, here we have a graph showing the general patterns of infection. If you look at this graph, of course, we have time across the bottom and virus production. We don't have to worry about numbers. And where you see the red bars, that's when the patient is symptomatic. And where you see the brackets, that's when infectious virus is being released. Up at the top, we see the typical profile for an acute infection. Virions are produced, symptoms occur, and infection is cleared. With persistent infections, the virion production continues for the life of this host. Symptoms may or may not appear just before death, depending on the virus. Infectious virus is produced throughout the infection. Now, if we're looking at latent reactivating viruses, the variation of persistent reactivation may or may not be accompanied by symptoms, but generally results in the production of infectious viruses. So herpes simplex 1 with the formation of cold sores is an example of this. And note that the person could be shedding virus even in the absence of symptoms. And then with a slow infection, you have periods of years intervening between a typical primary acute infection and the usually fatal appearance of symptoms. And HIV would be an example of this. And notice that even during that long period of time when symptoms are not obvious, the patient could be shedding virus. Primary infection with varicella zoster virus, or VZV, in susceptible individuals causes varicella, or chickenpox, which is usually harmless in healthy children whose immune system controls the infection. However, VZV establishes latency in dorsal root ganglia, and reactivation of viral replication and spread of the virus to the skin innervated by these neurons causes zoster, or shingles. Increasing age and compromised immune function are risk factors for complications of VZV infections. Viruses have been linked to approximately 20% of all human tumors worldwide. These transforming viruses encode viral oncoproteins that interact with cellular proteins to enhance viral replication. The transcriptional and post-transcriptional effects of these viral oncoproteins ultimately result in cellular transformation. Typically, these viruses encode specific oncogenes that promote cell cycle progression, inhibit DNA damage checkpoint responses, and prevent programmed cell death in an effort to promote viral propagation. And with this slide, this is just to show you that there's a wide variety of viruses that can cause transformation. Both RNA and DNA viruses are capable of that. Don't worry too much about that because we'll be talking about these viruses individually when we get further into the semester.